that I always share my experience from abroad, just to motivate the student, so that they also get to know that कि हाँ हम किससे किसकी टॉक सुन रहे हैं, ठीक है.
Yes, yes,
And this is
yes.
very important for the students also, so that they feel motivated and they, they learn something from you, uh, which of course they can learn from anywhere, but it is like their motivation that okay, if I have, maybe I have learned something new from abroad, that I can share here. And that would be my main core, like uh, bulk, or that I have to share here. And I would be bilingual during my presentation, just to make sure that I am like my feelings and my all flow of ideas and flow of information is smooth. Okay, so presently I am working as an assistant professor. My promotion is due, so I would be associate professor soon, in some months or maybe in years. It depends upon the committee that is taking care of all the promotion issues. And I did my PhD from Italy, postdoc from Canada, France. I had been the young scientist in Germany and the senior staff scientist in USA. This is what I always mention to my students, and also to all the scholars whom I get connected or get associated. Is very important for all these scholars to know that the most important thing that they should see when they want to know anything about any scientist in world. So this is my Google Scholar profile, showing about my citations, is showing about my H index and everything. So I have already got more than 100 publications, and in fact in this year only I have got 36 or 37 publications in this particular year, in 2024. So
Great.
I'm trying my level best to do some research. Like every 10 day I'm getting one of my paper accepted. And same thing, every day I'm getting my paper cited in at least two journals. Every day my papers, they are getting cited into others paper, two papers getting cited into others paper. Okay, so this is what it is important for the students also and the scholars to learn, that what are citation, what are H index. So this is my professional experience that has already been mentioned to you, and this is basically the area where I had been working. So I had worked in the field of genetics, also microbiology, molecular biology, then cancer biology, biochemistry, computational biology, then CRISPR. Also I worked in CRISPR. And then when I had been in USA, I was working in the field of biomarkers. So I would focus here in this research, um, in this research, like the talk I'm presenting here. So this is my first slide. I am sure Professor Gopalji, he knows about this slide. So I'm just sharing with you all, guys. Just see this slide and let me know what you could see from this particular slide. What message you are getting, what information you are getting from this particular slide. And anyone, they can just unmute yourself, or you can just raise your hand, then Professor Gopalji will allow you to just say a few words about this particular slides. That's it, and then we will move forward.
All are allowed to unmute anytime and speak.
Any answer from any scholars? Okay, so the thing is that no problem. I
एक ने कन्वी ने बोला मेल्स आर मोर रिकॉग्नाइज्ड आई गेस.
So,
यस,
hello.
रिया सिंह, यस बोलिए.
So here we can say that different people in a bunch in a group, there are different people with level like different genetic aspects.
Wonderful.
Yeah.
So you are more or less very close to the the heart of this information here.
Sir.
You are very close to the information that I was looking for. So basically, I just wanted to convey here that we are all part of a heterogeneous population. It means that in our population we have male, we have female, we have kids also, we have, aged peoples also, we have diseased people also. Okay, and we have different ages also, and also diseased people with different diseases. So there are some diseases that are, sex linked, there are some diseases that are age linked. So basically we are living in a population which is quite heterogeneous, but we all are unique and we all are different from each other. The main thing here is that when we consider ourselves, if we are diseased, it means that I am an individual, I am male, I am of this particular age, I am suffering from this particular disease, and my disease is unique in respect that I have a unique genome. Okay, so my genome is making me unique, and that's why my face is different from yours, and all my phenotypes and all my expressions are quite different from yours. So the thing is that this is what you can see from outside, but inside also when we have difference in genome. So say for example, Professor Gopalji and me, we are almost 99.99999% similar. But yes, there are some places where we have differences, and because of those differences only, Professor Gopalji looks like Professor Gopalji and I look like as Tikam Dakal. Okay, so this is the uniqueness that we all have. So how this uniqueness is related with science, how this uniqueness is related with disease, how this uniqueness is related with the medicines, how this uniqueness will to help us to develop the medicines, targeting one patient at a time, targeting the specific patient, or dealing with the specific individual. Okay, so this is what basically I mean to convey here. So, so we are talking basically about the cancer. I work mostly in the field of cancer, but sometimes I take also neurodegenerative disorders also and some other diseases. So, but more or less, I work in the field of computational and genome biology and computational biology and bioinformatics, and I do some lab experiments also. So previously, we used to have, for the treatment of cancer, we used to have traditional medicine that which includes radiation therapy, chemotherapy, then surgery. So these were the traditional therapies that you can see here. Uh, that you can see here. Okay. So, and if we talk about the precision medicine, because when I am talking about you, that you are quite different from others and you are unique in yourself, so it means that all the therapies that that should be targeted to you, and that should be like, given to you, should be unique for you, because you have a specific genome, you have a unique genome. So nowadays all our therapies, they are basically connected with the genetics, they are connected with the immunotherapies, they are connected with the targeted therapies. This is, we are talking about the cancer only. Okay, so the whole concept is called as precision medicine, and the treatment used is basically termed as personalized treatment. Okay, personalized treatment means treatment for individuals, specific individuals. Okay, means one, for one person there is one treatment, for other person there is another treatment. That is, for you the treatment would be different from the treatment that I would receive. Okay, so from here you can understand little bit more about the precision medicine and the personalized treatment. We can use this term interchangeably also. We can use here precision medicine, or we can say personalized medicine, that is more or less same. So here you can see on the left panel, you can see that we have three groups of patients, one in black, one in blue, and one is in light blue or olive blue. And for all these patients, we have only single drug, that is one capsule in yellow blue. Okay, and on the right side panel, you will see that yes, of course, here also we have three different groups: black group, blue group, and the olive blue group. But for these three groups of patients, we have different types of medicines. For one, we have capsules. For other, we have tablets also, and for others, they have mix of both. So the thing is that on the basis of genetics, we have to categorize the patients also. Earlier, the patients used to categorize based on their stages of cancer. Okay, this is breast cancer patient, and she is 40 years old, and she is 50 years old, she is 60 years old, like that. But nowadays, the most important thing is that, of course, we do have to consider the stages of the cancer also while making some remedial treatments modalities. But the most important thing here is that first we have to categorize the patients based on their genotype. And so, on the basis of the genotypes, we will have different patients, different groups of patients, and then we will use the targeted therapies for particular group of patients. So for one particular group of patients we will have different therapy, for another group of patient we will have different therapy. Okay, so this is basically the concept of personal precision medicine and personalized treatment. So my aim of this talk is really not to give you a lot of information and that you will just see here, you will just observe here, you will experience here, and then you will forget everything. So this is really not my aim, and I believe that Professor Gopalji has also included me, or he has brought me here just to, give you a thought that whenever you are attending any talk from any professor, you you should take some learning, or you should take some message home, that yes, of course, from this particular talk I learned these things. Okay, so this cancer biomarker area is a blooming area. It's like, upcoming area in the field of cancer research. And nowadays all the cancer research is based on biomarker discovery. Okay, so what basically biomarkers are? So here in the right side, left side panel, I have mentioned here that biomarkers are anything means that is related with our body, that can be measured as an indicator of a biological process. This means that there is some process going, there are many processes going in our system, our body, and if there is any indicator of process happening qualitatively or quantitatively, that indicator can be used as a biomarker. Okay, so biomarkers could be anything. They could be proteins, they could be cells, they could be DNA, they could be RNA, so they could be microRNAs, they could be some cellular components like mitochondria, exosomes, like that. Okay, so biomarkers could be anything. Basically, these are markers. They are giving us some information, and they are of biological nature. That's why we are mentioning them as biomarkers. Okay, so there could be different types of biomarkers. The most important biomarkers that I have mentioned here is, like, on the left side, we have predictive biomarkers and prognostic biomarkers and the diagnostic biomarkers. Okay, there are other types of biomarkers also, but when we are talking about the cancer and disease and related to treatment, related to disease progression, in cancer, we basically consider these three markers. So predictive biomarkers, basically these biomarkers, they are used to determine if an individual is more likely to experience a favorable effect from a medicine or not. It means that based on your genotype, we have selected a medicine for you. Okay, and we want to see if this particular medicine would give you a positive or favorable effect or not. So for that, whenever we will be giving the medicine, we will try to see if there is any gene that is going up or down, or is there any protein that started expressing. Means we will check for some biomarkers that is basically will give us information that, okay, if the patient would go, if the medicine would go good for the patient, you will have this biomarker going up or going down. So there will be an indication that this is a biomarker, this is a predictive biomarker, and whenever you are giving any medicine, so patient will experience the favorable effect only when this biomarker will go up, and this biomarker going up of this biomarker can be an indicative of a favorable response. This is basically predictive biomarker means. Okay, then we have the prognostic biomarkers. This indicates, the likelihood of disease recurrence or progression, whether this disease is going to happen again. We have cured the cancer, we have like undertaken the surgery, and after that there is a relapse of cancer. So for this we have the prognostic biomarker and the diagnostic biomarkers. You all know about the diagnostic markers. Basically these are the biomarkers that helps in the diagnosis of the disease. Yes, that okay, we checked for the qPCR of the COVID samples, and we got to know that yes, there is the load of viruses in your blood. Okay, so this is basically the diagnostic biomarkers. Okay, so there could be like the RNA coming from the viruses that has been checked by the machine, and if there is more of that particular type of RNA, we can say that yes, of course, there is a disease, there is virus, and we can say that we are we are suffering from this disease. Okay, so in cancer also there are some biomarkers that helps us in diagnosis of the disease, that okay, you have been diagnosed from this disease, you are diagnosed with this disease. Okay, means you are suffering from this breast cancer, so there will be some specific biomarkers for the breast cancer, there will be some specific biomarkers, diagnostic biomarkers for lung cancer, for and so on. Okay, so this is basically about the biomarkers. So we have 1 hour time, so I would go like slowly. I have many slides to show you, but I would skip some slides. I would be focusing more on some slides. So his reason is that my main aim is that you learn something from my slide, and you know, okay, we spent 1 hour with Dr. Tikam Chandakal, and we get to know about these things from his talk. Okay, now biomarkers, they can be classified in many ways. Okay, so here we have gene based biomarkers, we have protein based biomarkers, we have disease based biomarkers that I already mentioned. We have metabolic biomarkers also, we have some source based biomarkers also, we used to have some cellular biomarkers also. Okay, I have already got one paper published, and where I have it's published in 2024 only. It's about the biomarker research. So there I have mentioned that we have some cellular biomarkers also, where the cells can be used as the biomarker for the predictive uh, as a predictive biomarkers, or the prognostic biomarker, or the diagnostic biomarkers. So it's just skipping this. Okay, so who could be potential biomarkers? Now you know that okay, this is biomarker, this is a definition of the biomarkers, and biomarkers could be of different types, and biomarkers could be like gene based biomarkers, these could be protein based biomarkers. But the most important biomarkers that we deal with, or we deal uh, in context to cancer, are gene based biomarkers, protein biomarkers, SNP biomarkers, and RNA biomarkers like non-coding RNA biomarkers and regulatory regions. Okay, so anyone know about the non-coding RNAs?
पता होगा, बोल नहीं रहे हैं.
हाँ, आप लोग मुझसे इंटरेक्ट कर सकते हो, और कहीं भी कुछ दिक्कत हो तो आप
You
मुझसे
can
रोक
you
सकते
can
हो.
interact, you can ask question, you can reply the question.
हाँ, I really want to have this session in an interactive mode, so that
Yes.
we learn more things, and I would also feel happy that okay, I spent 1 hour, but I tried my level best to transfer my information, or the piece of information, or whatever knowledge I have. I am able to
Yes,
transfer to
निशीश
others.
शर्मा, अनम्यूट करके बोलिए.
Sir, uncoding biomarker, sorry, RNAs are those which doesn't translate any protein.
Good. Okay, so the main thing is that uh, of course, okay, thank you, you are right. The main thing is that we study about the central dogma, we study about the genes, we in these then these genes get transcribed into mRNA, and then mRNA get translated into proteins, that we all know. This is basically the center the main themes of the central dogma also. Now the genes, they have structure. Genes, they are not like, one gene after the another gene. There are many, the genes, they used to have promoters, they used to have stop regions also. They have 5 prime UTR, they have 3 prime UTR region. Okay. And our whole genome is 97% of the genome is basically non-coding, which is not getting translated into any protein. Only 3 to 5% of the DNA is getting transcribed into mRNA and then into the proteins. Okay. So you can see that we have almost more than 20 or 30,000 genes or proteins in our body, in human body. Okay. So if we consider, like, if a person is diseased, so in that case we may have some, defective proteins also getting expressed by our cells. In that case, we will have, the number would increase. So you can see that we have 30,000 genes, 30,000 genes present in our body. So, and now you can also see, that these 30,000 genes, they they are implicated in different diseases in different combinations. So for, if you start making the combinations of different genes, uh, 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 like, implicated into some different processes, there would be many different combinations that can come. Okay. So the main thing that I would like to convey here is that the 97% part of the genome is basically non-coding. But previously, this part used was considered as the junk DNA that you all already know. And nowadays, this DNA is not considered as the junk DNA. But in fact, now started scientists, they have started realizing that this part of the genome, which is not getting translated into protein, is is of regulatory nature. It means that this contain the information that regulate the 30,000 genes. Okay. So how the genes, they will behave, would be controlled to some extent by the non-coding regions also, first thing. And if we talk about the cancer, then we we basically talk about two types of genes. One, oncogenes. Basically, the oncogenes, they comes from the proto-oncogenes. And proto-oncogenes, they used to be the normal genes. But when there is like some, you know dysregulation of some any system inside our body, these genes become some proto-oncogenes, and then they become the oncogenes. So similarly, we used to have tumor suppressor genes also. So if we are talking about the cancer genes, then we have oncogenes and the tumor suppressor genes. And these non-coding RNAs, they control the expression of these oncogenes and the tumor suppressor genes also. Okay. So there are many ways in which these RNAs, these non-coding RNAs, they can regulate the disease. They can regulate the expression of the genes, and thereby they can regulate the progression of the disease or onset of any disease, or they can make the disease worse. They can, make the cancer go metastatic also. So, and they can also like, make the relapse of the disease. So it means that there are many non-coding RNAs, and in a particular disease, the RNAs, this non-coding RNA, they also express. They express in the sense they are getting transcribed, but they are not getting translated. Basically, the thing is that they are genes, but they are not getting converted into the protein. So they are DNA sequence that are getting transcribed. So it means that they are getting expressed, they are getting transcribed, but they are not getting translated into proteins. Okay, that's why they are considered as non-coding. So these non-coding RNAs, basically, they control. So in case of any particular disease, or in context to any disease, the different types of RNAs would come. In other disease, the RNAs would be different. In another disease, the RNAs would be different. For a particular disease and different stages, the RNAs may be different. Okay. So it means that in particular, in some disease, a particular RNA can be considered as the prognostic marker or a predictive marker or a diagnostic marker. It depends. But yes, of course, these RNAs can also be considered as the biomarkers. Okay. So I would not go more into details. So here, so if we want to consider, if you want to consider genes as the biomarkers, so what experimental approach that we would need to adopt to find genes as a potential biomarker for the cancer? Okay, so here we go. We have, if we have some samples and we want to know what are the gene biomarkers for this particular disease, then we can go for gene expression profiling, we can go for network analysis, we can go for survival analysis, and there are many other techniques that we can go for. And then we can find out that, okay, these are the genes that can be used as the potential biomarker for this particular disease. Okay, so how we do, I would mention in my, like, coming in next slides. Skipping this. Okay, so I'm like, giving you some uh, snapshots of my research that I'm doing in different cancer. So this is first uh, you know theme of my research, or you can say this is one research that I'm doing in my lab. It's a genome-wide transcriptomic profiling analysis of PC, pancreatic cancer. And here what we discovered a gene GSK3B as upregulated gene. Okay, it means that in case of pancreatic cancer, this gene is getting upregulated. So how we get to know that this gene is getting upregulated? Okay, if this gene is getting upregulated, it means that if we check for patients, and if we check in the patients, if we check for this particular gene, or if this gene is getting upregulated, we can say that this particular patient may get pancreatic cancer. Okay, so this is how we have to link the things. So the GSK3B gene can be used as a diagnostic marker for the pancreatic cancer patients. So here you can see that here you can see that we have taken the gene expression data. Gene expression data, it means that we have the pancreatic cancer patients data, um, the genes that are expressing in the pancreatic cancer patient, and the genes that are expressing in normal patients. Okay, so when we analyze this data, we get to know that this GSK3B gene, and there are some other genes also, but this was the most important genes for us. So we conducted further experiment with this gene, and we found that this GSK3B gene, it is basically implicated in pancreatic cancer. This is getting upregulated in pancreatic cancer patient. So in order to identify genes or genes biomarkers in relation to any disease, we have to go for the gene expression analysis. So that same we did here, and the same I conveyed here in my slide also, that if we want to identify genes as the biomarkers, then we have to go for gene expression profiling. Okay, so we took the gene expression data. There is a website, it is Gene Expression GEO website, where you can find all the expression data. Then we used to do some bioinformatics. I do this using R Studio is a language. Basically I use R Studio, and then we can find out the genes that are getting upregulated or downregulated in context to any disease. Here in our case, it's a pancreatic cancer. Okay, so we performed the genome-wide transcriptomic analysis to identify potential therapeutic targets or potential genes that can be, in our case, this was something that we wanted genes that can be targeted for therapies. Okay, so the teriflunomide was the drug that had shown positive response in case of pancreatic cancer patients. And then when we found that this GSK3B gene is getting upregulated, We just wanted to know what, and this GSK3B gene is not good for the cancer. So, and it is getting high upregulated. We just wanted to see if this drug is interacting with this particular protein or not. So based on this, in silico work or the expression data from the databases, we get to know that this GSK3B gene is highly upregulated in pancreatic cancer.
निश्चित शर्मा सर,
Yes, sir.
निश्चित शर्मा क्वेश्चन है. Can you unmute निश्चित शर्मा and ask question?
Sir,
Yes.
like we identify the function of gene by switching off or on its activity.
एक
So,
मिनट, yes, wait.
Ravi Tandon sir, welcome.
Am I visible?
हाँ.
Okay, so yes, please ask me question. I couldn't hear.
sir, like we identify gene functioning by its switch on or off its regulation activity. Sir, is there any another way to identify this disease is caused by this gene without dysfunctioning its activity?
I couldn't get your point clearly. Or maybe you can brief in Hindi, in your language, where in which you feel free to express your feelings also. So just
Uh,
mention everything clearly. What really you want to say to me?
Sir, like if anyone having disease related to genes, we
Okay.
can identify by switching off its functioning.
Okay. Perfect.
Then, sir, is there any alternative way to identify without switching off its activity?
Okay. The
Gene
thing is that
knockdown.
now this is what I am showing here. So you really do not know which gene is basically implicated in any particular disease. Okay, so in that case, what you have to do, you have to take the samples of the disease patient and the normal patient. Then you go for the transcriptomic analysis. Transcriptomic analysis, it means that you will get the gene expression data. It means that which gene is getting more expressed or less expressed in disease patient with respect to the normal individual. Okay.
तो
And
क्या वो डाटा हर डिजीज का अवेलेबल है?
सर, इन जीन एक्सप्रेशन जीईओ वेबसाइट जो है, वहां बहुत सारी डिजीज का बहुत तरीके का डेटा ऑलरेडी अवेलेबल है. हमें सिर्फ उस डेटा को हमारे काम के हिसाब से उसको एनालाइज करना है. अगर हम ये चाहें, से फॉर एग्जांपल, जैसा अभी इन्होंने क्वेश्चन पूछा, ये किसी एक पर्टिकुलर डिजीज का देखना चाह रहे हैं कि ये क्या वो पर्टिकुलर जीन किसी पर्टिकुलर उस एक पर्टिकुलर डिजीज में इम्प्लीकेटेड है या नहीं है, तो ये क्या कर सकते हैं, उस डिजीज से रिलेटेड जो एक्सप्रेशन डेटा वो चेक कर सकते हैं और फिर उस एक्सप्रेशन डेटा को एनालाइज करके और फिर अपनी जीन को क्वेरी में डाल के ये चेक कर सकते हैं कि क्या उन टॉप जीन्स के अंदर जो अप हो रही है और या डाउन एक्सप्रेस हो रही है, डाउन रेगुलेटेड हो रही है, क्या उसके अंदर उनकी जीन आ रही है या नहीं है. ठीक है. अब दूसरा चीज इनका कि अगर हम अगर हमें ये पता चल जाएगा, से फॉर एग्जांपल अगर हमें पता चल गया कि इस पर्टिकुलर डिजीज के अंदर ये जीन्स इंप्लीकेटेड है, तो हम क्या कर सकते हैं, फंक्शनल एनालिसिस कर सकते हैं, जिसके अंदर हम लोग उस पर्टिकुलर जीन को क्या कर सकते हैं, हम लोग नॉकडाउन कर सकते हैं या हम अप रेगुलेटेड या डाउन रेगुलेटेड कर सकते हैं उस जीन को. ठीक है, बाय ट्रांसफेक्शन एक्सपेरिमेंट्स. ठीक है. और उसके बाद में वी विल सी कि अगर वो जीन से फॉर एग्जांपल ऑन्कोजीन थी और हमने उसको डाउन रेगुलेट किया, तो क्या उससे फेवरेबल इफेक्ट मिल रहा है या नहीं मिल रहा है कैंसर पेशेंट को. इस तरीके की फंक्शनल स्टडीज हम लोग कर सकते हैं. वो तभी कर पाते हैं जब हमें पता हो कि इस पर्टिकुलर कैंसर के अंदर ये जीन इंप्लीकेटेड हो रही है. ठीक है. ब्लाइंडली हम लोग ऐसा नहीं कर सकते हैं कि हमें लग रहा है कि शायद इस जीन का हो सकता है, हमने उस जीन को का नॉकडाउन कर दिया और नॉकडाउन करने के बाद में उसका फंक्शनल एनालिसिस किया और पता लगा कि नहीं कि ये जीन कैंसर में रियली में हेल्पफुल है या नहीं है. नहीं, वो नहीं कर सकते हम लोगों को एक सिस्टमेटिक अप्रोच अडॉप्ट करनी पड़ेगी. और द सिस्टमेटिक अप्रोच इज दैट इफ वी वांट टू आइडेंटिफाई एनी जीन having role in any particular disease, then we can go for the expression data. We have to analyze the expression data. Then we will get to know about the genes that are getting upregulated, that are getting downregulated, and then we will try to see if our gene that was in our mind is in the list or not. Okay, so this is how we have to conduct the research. So I think I explained myself clearly and was helpful to the student also.
Yes, sir.
So
Thank you.
Yes.
okay, and in any case, if you have more queries, you will at in the end of my slide you will have my email ID also, so you can ask me questions also. Okay, so this is not a problem. So then we what we did, we took this protein, GSK3b protein, and we just did some docking experiment with molecular dynamics and simulation, everything, and we found that yes, there is the active site, and this drug is basically binding to this active site, and in this way this is making this protein non-functional in cancer patient, and this could be the reason why we were experiencing the favorable effect from this drug. Okay, so likewise we can do the same approach can be adopted for other cancers also, of course with a different kind of drug and with different kind of therapeutic targets, gene therapeutic targets or the protein targets. There was there is another research work that I already conducted, and this is already published also. Here also, we our aim is to identify biomarkers for a particular disease. Okay, so the story was something different, and then we got to know something relevant in context to cancer. Basically, we were working with the ape1 protein, APE1. It's basically called as ape1 protein, and this protein had has the function in DNA repair. Okay, so not I would not go into the more details. I would come directly here. Then what we did, we we wanted to see the interacting partners of this ape1 protein. Means in the cell to which other proteins this ape1 protein is also interacting. Okay, so interestingly what we found is that there is a protein, it is called as PRDX1. This protein is basically involved in redox balancing the redox status of the cell. So our this ape1 protein, it 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 interacts with the PRDX1 protein. This we got from the our mass spec analysis. So then we got interested in this interaction, that okay, this ape1 protein is basically a protein having role in DNA repair, and this PRDX1, it has completely different role in the cell. So there is no relation between the you know the the ape1 protein and the PRDX1 protein, and this was really very interesting for us, that why these two proteins are interacting. So one thing that was you know that was very interesting is that this ape1 protein has a redox active region, and region was redox active region, because this at this in this region, this ape1 protein had some redox active cysteine residues, and this was the link that helped us, that yes, of course, if this ape1 has redox active cysteines, it means that it can reduce the other proteins or getting oxidized like that. Okay, so then we started working on this particular ape1 and the PRDX1 protein, and what we did, we down regulated the PRDX1. Okay, so what in case if we do not have PRDX1, a protein which has role in maintaining the redox status of the cell, then there is upregulation of the IL-8. But the main question was that how this IL-8 is getting up regulated. It is highly expressed in the cells where we do not have the PRDX1. So then we did some other experiments also. In those experiments we get to know that when when we have the PRDX1, functional PRDX1, the ape1 is resides in the cytoplasm, and when we have the knockdown of the PRDX1, the ape1 goes to the nucleus, and then it is doing something there. Okay, so what the ape1 doing in the nucleus is a story which I will be showing here. But the main thing is that in the cytoplasm, this PIDX1 was interacting with the ape1, and when there is no PIDX1, it means that now the ape1 is free. So the main aim of the ape1 is to go inside the nucleus. It is doing role in the in the in the DNA repair also. But at the same time, we found that this ape1 has some cysteine, redox active cysteine residues in the N terminal, and this ape1 is causing the reduction of the NF kappa B, and thereby it is making the islet gene to express more. Okay, so we get to know from this particular study that PIDX1 can be used as a biomarker. If there is low expression of the PIDX1, we can say that we may get some cancer disease. In also, islet gene, which is a pro-inflammatory chemokine, and if we see that if a patient is expressing more of the islet gene, we can say that this patient may get cancer in later stage of his life. Okay, so like this we used to do research like this. We used to we used to find out the genetic or the gene biomarkers or the protein biomarkers in relation to, in context to any particular disease. So now we talk about the SNP biomarkers. Let me check the time. Okay, so we are almost close to end of the talk also. I have
नहीं,
many
10-15
slides.
मिनट ज्यादा भी होगा तो कोई बात नहीं है,
I
स्टोरी मच.
Yeah, I have many slides, but I would mention a few more slides just to explain that yes, when we want to identify genes as the biomarkers, what experimental methodologies we have to adopt. When we want to see or we want to identify protein as the biomarker, what the experimental approach we have to identify. And now we are talking about the SNP biomarkers and how these SNP biomarker could be used as the biomarker for the cancer to identify for the diagnosis of the disease, for prognosis of the disease, or for for predictive response, checking the predictive response. So this, we all know that here in the left side, here we can see that this is the coding region, this is the regulatory region, this is basically a gene, and this coding region is getting you know is getting translated. This is the coding region of the gene. It means that it is basically the gene which is getting transcribed and also getting translated into protein. So the SNPs could be of different type. Some SNPs, they are non-coding SNPs. It means that these SNPs, they are found in the regulatory regions and they are not found in the coding region. If there is an SNP which is found in the coding region, it would be considered, it would be termed as coding SNPs. It means that if there is change in the gene, the change we will see in the protein also. In exceptional cases, because for some because of the wobble hypothesis, a wobble effect, if there is a change at the third position, for some amino acid there used to be no change. Of course, we know all about this thing. Okay, we have 64 combinations of amino acids. So the codons, they are coming and they are coming together and they are coding for a particular amino acid. So if there is a genetic change or the change in the third codon, maybe we can see the effect, maybe we cannot see the effect. But if there is an SNP in the coding region, that SNP would be termed as coding SNP, and that coding SNP may have structural and functional implications or effects on the protein it is related to. Okay, so this used to be a strategy to identify SNP biomarkers. So first we have to screen the citation, we have to identify genes, then we have to selection of the genes, then selection of the transcripts. And that I mean to say here, okay, maybe I can skip here and I can show here like this. So I started finding SNP biomarkers of the IL-8 gene. Okay, so what I did basically is that I went to the database, I checked the database, I explored the database, and I get to know that there are many SNPs associated with this particular gene, and there were 41 coding SNPs, 41 coding SNPs present in this particular gene when I conducted this disease. Maybe now maybe the number must have already increased. So there were 41 coding SNPs. So I analyzed the 41 coding SNP because I wanted to see if there is any genetic change at the DNA level. So same is we can see, or what we see when we see that change in a protein. Okay, so it is basically the coding, if the effect in the protein. So there we identify these SNPs, and we identified the SNPs. Those were called as non-synonymous SNPs. It means that SNPs having that basically change the amino acid in the protein. Okay, then this is the figure from one of my paper got published in scientific reports, and this was the first study. I would say this was the first study wherein I, in my opinion, I was the first person to make this approach to identify the SNP biomarkers, or to identify the pathogenicity of any SNP related to any gene. Okay, तो कहने का मतलब बेसिकली ये था कि एक जीन है उसके अंदर उसके उस जीन के अंदर काफी सारे म्यूटेशंस हो रखे हैं, बट उनमें से कौन सा म्यूटेशन ऐसा है जो असली में डेंजरस है, जिसको हम लोग क्या कहते हैं, पैथोजेनिक कहेंगे, या डैमेजिंग कहेंगे, या डिलीटियस कहेंगे. तो पैथोजेनिक म्यूटेशन है या नहीं है, उसको आइडेंटिफाई करने के लिए हमने ये पूरी स्ट्रेटेजी बनाई थी, जिसके अंदर हम लोगों ने टूल्स यूज किए थे, जैसे सिफ्ट और प्रोवियन. ये बेसिकली वो टूल्स है जो एसएनपीज की का प्रेडिक्शन करते हैं कि क्या ये एसएनपी रियली में डेंजरस होगा नहीं होगा, और उसके लिए हम लोग क्या करते हैं, सीक्वेंस होमोलॉजी बेस्ड अप्रोच यूज करते हैं, जो होती है सिफ्ट एंड प्रोवियन, जो होते हैं वो सीक्वेंस होमोलॉजी बेस्ड अप्रोच के ऊपर काम करते हैं. इसी तरीके से एक पॉलीफेन आता है, वो क्या करता है, स्ट्रक्चरल होमोलॉजी बेस्ड अप्रोच के ऊपर काम करता है, और ये बताता है कि वो एसएनपी जो है वो असली में डैमेजिंग है या नहीं है. ठीक है, क्या वो जो एसएनपी है वो न्यूट्रल तो नहीं है. और बहुत बार ऐसा भी होता है कि अमाइनो एसिड चेंज हो रहे हैं, बट असली में प्रोटीन के स्ट्रक्चर में कोई चेंज नहीं हुआ, प्रोटीन के फंक्शन में कोई चेंज नहीं हुआ. सो ये हम लोग यहाँ क्या कर रहे थे, ये जो टूल्स हैं, इन टूल्स की हेल्प से हम लोग वो आइडेंटिफाई करना चाह रहे थे. तो हमारे पास में कितने थे, टोटल 41 एसएनपीज थे. उस 41 एसएनपीज में से हमें 8 एसएनपीज ऐसे मिले जहां हमें क्या मिला कि उन एसएनपीज की वजह से जो चेंज हो रहा था, वो चेंज होने से जो आइलेट प्रोटीन है उसका स्ट्रक्चर चेंज हो रहा है. देखिए, स्ट्रक्चर, किसी भी चीज का स्ट्रक्चर चेंज होता है पहले, उसके बाद में फंक्शन चेंज होता है. मैं इसको एक एनालॉगस तरीके से कर सकते हैं कि हमारे पास में 2 पैर हैं, चीता के पास में 4 पैर होते हैं. ठीक है, और चीता हमसे तेज दौड़ पाता है क्योंकि उसके अंदर स्ट्रक्चरल डिफरेंसेस है. हमारे रनिंग की जो मशीनरी है और उसकी रनिंग की जो मशीनरी है, उसके अंदर स्ट्रक्चरल चेंजेस है. ठीक है, एक बर्ड है जो उड़ सकती है क्योंकि उसके अंदर जो बर्ड के उड़ने की जो मशीनरी है उस, और हमारी जो है वो उसके अंदर चेंजेस हैं. ठीक है, तो स्ट्रक्चर बहुत इंपॉर्टेंट रहता है. अगर किसी प्रोटीन का स्ट्रक्चर डिस्टॉर्टेड हो रहा है तो उस प्रोटीन के फंक्शन के अंदर चेंज होना बहुत प्रोबेबल रहेगा. ठीक है, अब हमने फिर क्या किया, एक ही एनालिसिस का बहुत अच्छा एनालिसिस है ये. इसको कौन सा एनालिसिस हम लोग कहते हैं. इसके अंदर हमने क्या किया कि ये हमारा आइलेट जीन है. ठीक है, ये आइलेट जीन है. हमने इसके अंदर क्या चेक किया कि ऐसे कौन कौन से अमाइनो एसिड्स हैं प्रोटीन के अंदर जो हाईली कंजर्व्ड है, और ऐसे कौन से जो लेस कंजर्व्ड है. सी, अगर किसी प्रोटीन के अंदर कंजर्व का मतलब क्या है, इवोल्यूशनरी कंजर्व, कि एक प्रोटीन जो जब से मैमल्स हुए तब से वो प्रोटीन है, और उस प्रोटीन के अंदर एक पर्टिकुलर अमाइनो एसिड है, वो पहले भी एलेनिन था और आज ह्यूमंस में भी एलेनिन है, चिंपैंजी में भी एलेनिन था, मंकी में भी एलेनिन था, ठीक है, मींस वो हाईली कंजर्व्ड हो गया है, वो अमाइनो एसिड उस पर्टिकुलर प्रोटीन के उस पर्टिकुलर पोजीशन के ऊपर कंजर्व हो गया है, ठीक है। और कुछ ऐसे होंगे कि आइलेट प्रोटीन चिंपैंजी के अंदर थोड़ा सा चेंज है और मंकीज के अंदर फिर अलग है, रिशियस मंकीज के अंदर अलग, बिल्कुल अलग, कुछ के अंदर और के अंदर गोरिल्ला के अंदर अलग है। तो हमने क्या किया, एक एनालिसिस किया जिसे कंजर्वेशन एनालिसिस बेसिकली है, कि उस पर्टिकुलर प्रोटीन के अंदर हर एक अमाइनो एसिड इवोल्यूशनरी कितना कंजर्व्ड है वो हमने कलर कोडिंग से बताया है। अगर 9 है तो हाईली कंजर्व्ड, और अगर 1 है मींस कलर कोडिंग में, मतलब अगर ये वाला कलर शो हो रहा है इट मींस वो जो अमाइनो एसिड है वो वेरिएबल पोजीशन है। अगर जो वेरिएबल है और वहां अगर म्यूटेशन हमें दिख रहा है और उस म्यूटेशन से वो पर्टिकुलर जो वेरिएबल अमाइनो एसिड है वो चेंज हो रहा है, तो उसका कोई इफेक्ट नहीं होगा प्रोटीन के ऊपर, क्योंकि वो तो अमाइनो एसिड चेंज होता ही रहता है। वो अमाइनो एसिड जो कभी चेंज नहीं होता है और किसी म्यूटेशन के कारण ह्यूमन के अंदर चेंज हो गया है, तो आप मान के चलिए कि वो चेंज होगा दैट वुड ब्रिंग सम स्ट्रक्चरल चेंजेस इन द प्रोटीन ऑफ आवर इंटरेस्ट, और उस स्ट्रक्चरल चेंजेस की वजह से प्रोटीन के अंदर फंक्शनल चेंजेस भी हो सकते हैं। दिस इज वेरी प्रोबेबल, ठीक है। तो आपको जैसे मैंने ये बताया था कि जैसे E31 पोजीशन है, जिसे E से A हो गया, E से K हो गया, S41 हो गया, I55 हो गया, है ना। तो हमने देखा E31 के ऊपर 31 पोजीशन के जो ऊपर E है वो हाईली कंजर्व्ड है, ठीक है। इसी तरीके से हमने 50 कौन सा था, एक और था 41 के ऊपर जो देखा हमने वो ज्यादा कंजर्व नहीं था, ठीक है। उसका 59 और 70 है, 59 देखा तो 59 वाला बहुत ज्यादा कंजर्व्ड है, ठीक है। तो इससे हमें ये पता चला कि ये जो पोजीशंस है इसके ऊपर जो चेंज हो रहा है कि ई की जगह के हो गया और ई की जगह जो ए हो गया, उसकी वजह से ये हाईली कंजर्व्ड अमाइनो एसिड है वो चेंज हो रहा है। और ये हाईली कंजर्व था इवोल्यूशनरी पॉइंट ऑफ व्यू से। तो इसका मतलब था कि ये अमाइनो एसिड का ये रेसिड्यू जो है इस पोजीशन के ऊपर ये इंपॉर्टेंट था, और अब चेंज हो गया है तो उसके कारण कुछ ना कुछ स्ट्रक्चर्स के अंदर चेंज आएंगे और फंक्शन के अंदर चेंज आएंगे। तो ऐसे करके हम इन अमाइनो एसिड्स को इस पर्टिकुलर पोजीशन के ऊपर पैथोजेनिक बोलेंगे, कि ये इसका इफेक्ट डिलीटीज होगा, पैथोजेनिक होगा। अब इस सिनेरियो को समझिए आप, कि अगर हमने जेनेटिक स्क्रीनिंग करी और हमें पता चला, और हमें पता है पहले से बेस्ड ऑन आवर पहले की जो हम लोगों ने इतनी सारी इंफॉर्मेशन गैदर कर रखी है, और हमें पता चल जाए कि E31 पोजीशन के ऊपर आइलेट के अंदर ई की जगह के आ गया है या ई की जगह ए आ गया है, तो हम बोल सकते हैं कि ये जो प्रोटीन के अंदर जो डिफेक्ट हुआ है उसकी वजह से जरूरी आगे फ्यूचर के अंदर कोई ना कोई प्रॉब्लम कैंसर से रिलेटेड इस इंडिविजुअल को रह सकती है, ठीक है। तो इस तरीके से ये जो इंफॉर्मेशन होती है एसएनपी वाली इंफॉर्मेशन या जीन्स की इंफॉर्मेशन या प्रोटीन से रिलेटेड इंफॉर्मेशन, प्रोटीन का ज्यादा बनना कम बनना, जीन्स का ज्यादा एक्सप्रेस होना कम एक्सप्रेस होना, किसी म्यूटेशन का होना, किसी एसएनपीज का होना, वो भी ऐसी जगह पे होना जो हाईली कंजर्व थी, उससे ये पता चलेगा हमें कि हम इन एसएनपीज को एज अ बायोमार्कर यूज कर सकते हैं, ठीक है। और ये फिर हमने फाइनली फिर स्ट्रक्चरल बायोलॉजी करके हमने बताया कि असली में ये जो भी हमने जो ये कंजर्व रीजन वाले थे अमाइन रेसिड्यूज, वो इस पर्टिकुलर प्रोटीन का दूसरे प्रोटीन के इंटरेक्शन में काफी इंपॉर्टेंट रोल प्ले करते थे। और अगर वहां चेंजेस होंगे तो फिर वो इंटरेक्शन नहीं होगा, और उसके कारण फिर क्या होगा कि उसका जो फंक्शन है उस स्ट्रक्चरल चेंजेस के कारण उस प्रोटीन के फंक्शन के अंदर भी चेंजेस आ गए। ये ऑलरेडी हमने पब्लिश कर लिया था, ठीक है। सेम तरीके का काम हम लोगों ने लंग कैंसर के अंदर किया। उसके अंदर भी हमने जेनेटिक वेरिएंट्स जो थे उनकी स्टडी करी और वो पब्लिश किया, एक अच्छा जर्नल है जीनोमिक, उसके अंदर पब्लिश किया था। आप लोग इंटरेस्टेड हो तो इस पेपर को जाके देखिएगा, आपको और ज्यादा अच्छी इंफॉर्मेशन मिलेगी और सारी चीजें डिटेल में मिल जाएगी, ठीक है। कुछ चीजें स्किप कर लेता हूं मैं, ठीक है। यहां आता हूं। अब जैसा कि मैंने आपको आरएनएस के बारे में बताया था, नॉन कोडिंग आरएनएस के बारे में। सो नॉन कोडिंग आरएनएस के अंदर वी हैव ऑफ कोर्स वी डू हैव लाइक अ
और कितने स्लाइड हैं?
हेलो,
हाँ, कितने स्लाइड है? स्लाइड्स कितने? मेरी आवाज आ रही है?
हाँ हाँ आ रही है।
कितने
सर कुछ
स्लाइड है वो बोल रहा हूँ कि और कितने देर लेंगे?
मेरे मैक्सिमम 10 मिनट
ठीक है, ठीक है, ठीक है
है
जी।
ना,
हाँ,
मैक्सिमम 10 मिनट सर, ठीक
ठीक
है।
है जी।
सो कुछ जो नॉन कोडिंग आरएनएस होते हैं उसके अंदर सर्कुलर आरएनएस होते हैं, एमआई आरएनएस होते हैं, एमआरएनएस होते हैं। अब मैंने क्या किया, देयर इज अ डिजीज एंडोमेट्रियल कैंसर कहते हैं उसको हम लोग, ये एक जेंडर स्पेसिफिक डिजीज है, ये फीमेल्स के अंदर होती है। यू जो इनका होता है ना, मतलब जो इनका यूटेरस वगैरह होता है उससे रिलेटेड कुछ डिजीज होती है ये, और इसकी कंप्लीटली एटियोलॉजी और पैथोफिजियोलॉजी वो बिल्कुल डिफरेंट होती है। तो पहले एंडोमेट्रोसिस होता है, फिर एंडोमेट्रियल कैंसर भी हो जाता है। तो पैथोफिजियोलॉजी बहुत कॉम्प्लिकेटेड होती है इस डिजीज की। तो हमने क्या किया, ये पेपर लास्ट टू लास्ट ईयर ही पब्लिश हुआ था। मैंने एक अप्रोच बनाई जिसके अंदर, जिसको मैंने कहा सर्कुलर आरएनए, एमआईआरएनए, एमआरएनए इंटरैक्टोम एनालिसिस। ओके, क्या कहने का क्या मतलब है इसके अंदर? इसका मतलब बेसिकली ये है कि कुछ सर्कुलर आरएनएस होते हैं, गोल शेप होते हैं, इसलिए उनको सर्कुलर आरएनएस कहते हैं। एमआईआरएनएस होते हैं, एमआरएनएस होते हैं। एमआरएनए हम सब जानते हैं। एमआईआरएनए क्या होते हैं? वो एमआरएनए की साइलेंसिंग के अंदर हेल्प करते हैं। एमआरएनए से जाके बाइंड कर लेते हैं, जैसे ही एमआरएनए डबल हो गया, डबल मतलब आरएनए जब भी डबल स्ट्रैंडेड हो जाएगा तो वो उसका क्या हो जाएगा, साइलेंसिंग हो जाएगा, मतलब वो उसकी चॉप कर देगा। रेस कॉम्प्लेक्स वगैरह के कॉन्सेप्ट आते हैं, जिसको हम लोग आरएनए साइलेंसिंग भी कहते हैं, ठीक है। तो कहने का सेंस क्या है कि एमआईआरएनए जो एमआरएनए को साइलेंस करता है, और सर्कुलर आरएनए है बेसिकली, वो क्या करता है, एमआईआरएनए के अंदर बाइंड, एमआईआरएनए को सीक्वेस्टर करता है, बाइंड करता है। और सर्कुलर आरएनए के काफी सारे रोल होते हैं, उसमें से एक रोल ये भी होता है कि वो एमआईआरएनए को सीक्वेस्टर करता है। सीक्वेस्टर कर देगा, इसका मतलब उसको पकड़ लेगा। इसका मतलब अगर उसने पकड़ लिया तो एमआईआरएनए अब जाके एमआरएनए के ऊपर बाइंड नहीं करेगा, और एमआरएनए के ऊपर बाइंड नहीं करेगा। तो अगर कोई ऑन्कोजीन है, अगर कोई ऑन्कोजीन है तो वो जीन का एक्सप्रेशन बहुत ज्यादा होगा फिर, ठीक है। मतलब उसका प्रोटीन जो बन रहा है वो बहुत ज्यादा बनेगा। तो बहुत बार इस तरीके के रेगुलेशन होते हैं, कब किस प्रोटीन का बनना है, कब किस प्रोटीन का नहीं बनना है, किस, और प्रोटीन बन गया, उसका काम खत्म हो गया, तो उसका उसको उस प्रोटीन को वापस से डिग्रेड करवाना, प्रोटेसोमल डिग्रेडेशन वगैरह होता है। तो काफी लेवल के ऊपर, ट्रांसक्रिप्शन लेवल के ऊपर, ट्रांसलेशन लेवल के ऊपर, पोस्ट ट्रांसलेशन लेवल के ऊपर काफी टाइप के रेगुलेशंस होते हैं जो जो हमारे पूरे सेलुलर सिस्टम को गवर्न करते हैं और उसको टाइटली रेगुलेट करते हैं कि कैंसर ना हो, ठीक है। तो हमने इस तरीके का एनालिसिस किया, जिसके अंदर हमने क्या किया, एंडोमेटल कैंसर के अंदर जो सर्कुलर आरएनए थे उनको आइडेंटिफाई किया। फिर हमने उसके अंदर ये देखा कि इन सर्कुलर आरएनएस के अंदर कौन कौन से एमआईआरएनएस की बाइंडिंग साइट्स है, ठीक है। तो उन मतलब उस सर्कुलर आरएनए की सीक्वेंस को लेके हमने ये मैच करने की कोशिश करी कि इसके अंदर, सर्कुलर आरएनए के अंदर कौन कौन से एमआईआरएनए बाइंड कर सकते हैं, मींस ये सर्कुलर आरएनए किन किन एमआईआरएनए को सीक्वेस्टर कर सकते हैं। और फिर एमआईआरएनए से हमें ये पता लगाने की कोशिश की कि ये एमआईआरएनए बेसिकली किस प्रोटीन्स को टारगेट करते थे, ठीक है। समझ में आ रहा है? तो इस तरीके से हमने पूरे एक इंटरैक्टोम एनालिसिस किया और ये बताने की कोशिश की कि एंडोमेटल कैंसर के अंदर ये दो सर्कुलर आरएनए थे, वो दो सर्कुलर आरएनए इस तरीके से एमआईआरएनए को बाइंड कर रहे हैं। फिर हमने ये बताया कि वो एमआईआरएनए किन प्रोटीन्स को टारगेट करते हैं, वो भी हमने आइडेंटिफाई किया। और हमने पता लगाने की कोशिश की कि क्या ये पर्टिकुलर एमआईआरएनए एमआरएनए वाला जो इंटरेक्शन है ये ऑलरेडी नोन है क्या एंडोमेटल कैंसर के अंदर केस में। तो पता चला यस है, किसी में नो। तो हमें पता चला कि ये जो एमआईआरएनए इस प्रोटीन को रेगुलेट करता है, इस प्रोटीन को डाउन रेगुलेट करता है बेसिकली, और ये अभी तक एंडोमेटल कैंसर में पता नहीं है। तो ये अपना आप एक नई स्टडी होगी आगे काम करने के लिए, ठीक है। तो इस तरीके से चीजें की जाती है। ये हमने पब्लिश कर दिया वर्क को। ये हमने पूरा बता रखा कि एंडोमेटल कैंसर के अंदर हम जो सर्कुलर आरएनए थे, वो सर्कुलर आरएनए एमआईआरएनए को कैसे बाइंड कर रहे हैं, एमआईआरएनए फिर कहां जाके किस किस प्रोटीन को कर रहा है और उससे क्या इफेक्ट आ रहा है, वो सारी चीज हमने यहां बता रखी है। ये भी पेपर पब्लिश हो गया था, बहुत अच्छा पेपर है ये, आप पढ़िएगा जरूर उसको, ठीक है। उसके बाद में बहुत सारी स्टोरीज हैं कि कैंसर के अंदर ड्रग रेजिस्टेंस होता है, कैंसर की हम लोग चिप भी डेवलप कर रहे हैं, ठीक है। माइक्रोफ्लूइडिक्स चिप मॉडल्स हैं बहुत बहुत तरीके के, आई एम वर्किंग ऑन दिस फील्ड आल्सो, ठीक है। और बाकी सारी चीजें मैं नहीं एक्सप्लेन करूंगा आपसे ज्यादा। ये कुछ मेरे स्टूडेंट्स के वर्क है जो जिसके अंदर भी हम लोगों ने एमआईआरएनएस को आइडेंटिफाई किया इन रिलेशन टू ब्रेस्ट कैंसर, और पेपर ऑलरेडी पब्लिश्ड हैं ये सब। ये जो कोविड के ऊपर मैंने एक थेरेपी निकाली थी, बहुत अच्छा पेपर है, ये भी आप पढ़िएगा अगर आपको मिले तो। इसके अंदर भी मैंने पूरी कंप्यूटेशनल बायोलॉजी का अप्रोच लगा के एक बहुत सिंगल ऑथर्ड पेपर छापा था और एक थेरेपी डिजाइन करी थी कोविड के लिए। ये बहुत अच्छे जर्नल के अंदर पब्लिश हो रखा ऑलरेडी। फिर मैंने कुछ वैक्सीन कैंडिडेट्स भी निकाले थे, ये भी मैंने ऑलरेडी सिंगल ऑथर पब्लिश किया था, ये भी अवेलेबल है। तो बायोमार्कर्स के केस के अंदर मैं आपको ये बताना चाहता हूँ कि डिफरेंट टाइप्स के बायोमार्कर्स होते हैं। हम टूल्स एंड टेक्निक्स से उनको आइडेंटिफाई करते हैं, फिर उनकी पोटेंशियल एप्लीकेशन देखते हैं एज अ प्रेडिक्टिव, प्रोग्नोस्टिक और डायग्नोस्टिक बायोमार्कर्स। फिर हम लोग चैलेंजेस कुछ फेस करते हैं और उससे हमारा फ्यूचर डायरेक्शन डिसाइड होता है कि हाउ वी कैन यूज और इंटीग्रेट
इस पर भी एक दिन ले लीजिएगा ना, एक आध महीने बाद।
जी सर, जी सर, जी सर,
जी,
जी।
अच्छा रहेगा।
तो इस तरीके से दीज आर माय कोलैबोरेटर्स जिनके साथ में मैं काम करता हूँ। कुछ काम चल रहा है और आगे काम करने हैं। ये मेरी छोटी सी लैब है, मेरी लैब में टीम मेंबर्स हैं मेरे ये।
बहुत
और आप